Now on to lipids. Now lipids are a different kind of macromolecule class because they do covalently join little bits together, but to form the biggest structure, there's not a lot of covalent bonds between them, and that's kind of cool. The basic unit is often drawn kind of like this, where you've got yourself a round circle and two lines. The circle represents the hydrophilic head, and that would contain polar groups that can interact well with water, like there's nitrogen areas in there, and phosphate, which has lots of oxygen, and oxygen has lots of charge for water to play with. And then these red lines here represent the fatty acid tails. They would be hydrocarbons with very few polar or charged groups. That means that they have different chemical properties. This loves to play with water, hydrophilic, and these are hydrophobic. They don't mess with water very well. So these are formed by having different units. We'll have a glycerol, which is a 3-hydroxyl containing 3-carbon group, and to 2 positions on those hydroxyls we'll have the fatty acid tails, and on the 3rd hydroxyl on the glycerol we'd have a larger polar group attached. So the glycerol is kind of the intersection for all these interesting elements. Now this will all be covalently bonded, so it won't fall apart in any way, and it will join the larger group right here. We can see lots of its friends down there, and this would insert into the membrane with its polar head sticking up and the non-polar tails interacting with each other. Now they're interdigitating, they're locking in with each other. That's the way the model works, and it's not the way it is in reality. But what you can see with this arrangement is that at the top, outside of the cell, if this was like the surface of an organism, there would be water in the environment, an aqueous environment. And down here, where it's kind of turbid with some sort of cloudy stuff in there, that represents the intracellular environment, still aqueous, but with proteins and all of the different solutions of salt and such available for life's work to behave. The way that this membrane works is it spontaneously aligns all of the top polar groups towards the aqueous outside and the bottom hydrophilic groups towards the aqueous center of the cell. And the hydrophobic tails are able to interact with each other in such a way that they'll hold it together in sort of a sheet. So imagine this extending forward and backward and to either side to form a covering around the outside of the entire cell. Now, this is the physical structure, the makeup of a membrane that organizes things. These aren't catalytic. They don't do any real work. There are exceptions, I'm sure, but in this case, they sort of form the foundation for stability. Here we could see if a protein was built inside the membrane, and this will represent my protein, this tail is to keep me from getting my fingers in the mineral oil, it kind of can work its way inside of the membrane. Now, proteins don't dodge in and out of a membrane. It doesn't work that way. But what you can see is that the membrane can accommodate all of the movements. It'll spontaneously allow that to fit together. Now, if this was a protein that was embedded in a membrane and it would be put there by the rough endoplasmic reticulum, which would build the protein in place in the membrane, the protein will probably have a hydrophilic top part and a hydrophilic bottom part, and all the parts that are found in the red region would be hydrophobic, with amino acids that have little to no charge or are small and work well with the hydrophobic environment. So this is how the general structure of a membrane works, with the individual phospholipids aggregating together with their hydrophobic fatty acid tails, the red parts, to create a larger structure that forms the basis for lots of different types of organelles and membranes and cell surface structures and so on.